Hello, can you hear me?
Hello, Professor Mirbolooki. Yes, we could hear you, and
Cool.
we will be starting in a few minutes.
Sure, thank you.
Thank you very much. Well, hello to everyone, and a very good evening or good night to wherever you are watching us. My name is Dr. Ehsan Sharif Pagala, and I'm glad to be hosting this event tonight. So briefly, before me introducing our special guest speaker, I would be just briefly explaining about TPCF, so Tehran University of Medical Sciences Preclinical Core Facility. We're a center where we're providing various different imaging modalities and providing scientific services to various different researchers. As well as that, we're providing various different sort of educational sessions, such as this from TPCF webinars, which we are sort of planning to having these sort of monthly basis. And so it's an honor to have our, I think it's probably sixth or seventh speaker, Professor Mohammad Reza Mirbolooki, and it's an honor to have him here with us tonight. So briefly about Professor Mirbolooki, he's a professor of anatomy and physiology, as well as he's an undergraduate department chair at Southern California University of Health Sciences. And his research interests are obesity and diabetes. In particular, Professor Mirbolooki is interested in pancreatic islets and brown adipose tissue and the critical roles in glucose homeostasis. So the title of Professor Mirbolooki's talk tonight is Preclinical Imaging of Brown Adipose Tissue and Pancreatic Islets. Professor, it's an honor to have you, and I'm very glad and very eager to listen to your amazing talk. Platform's all yours.
Thank you so much for this warm welcome and introduction. I was really glad to see that we have such a center in Tehran University. And it's like a big progress for Tehran University to have all this facility and have a preclinical imaging in there. So congratulations to you and your team. And thank you so much for doing this. So let me share my screen. Okay, so I think that you should be able to see my screen now.
Yeah.
Yes. Okay, so today I'm going to talk about two things. One will be the brown adipose tissue and imaging of brown adipose tissue in rodents, and then briefly about also pancreatic islets. So my dissertation of my PhD was about pancreatic islets, but as a postdoc, I was working both on pancreatic islets and brown adipose tissue at UC Irvine here. So if I want to explain a little bit about brown adipose tissue, it's a tissue that it's known for its thermogenic activity. So they have a critical role in burning basically fats and carbohydrates. And there are lots of papers that talk about that they can be a good target combating obesity and diabetes, knowing about this extraordinary metabolic capacity they have because of the mitochondria, lots of mitochondria that they have. And then there's a protein in the mitochondria that they have. You can see it here, which is called UCP1. And it's uncoupling protein 1, basically wastes the hydrogen ions in the inner membrane space of the mitochondria. So these hydrogen ions are not used to create ATP. So when these protein express more in there, although the cell gets the glucose, gets the lipase, gets free fatty acids, go to the acetyl-CoA and the Krebs cycle, then still will not be able to create ATP because the amount of the hydrogen ion that goes in there will be wasted. It's not going to go through ATP synthase and makes the ATP. So that's the whole idea. And because of that, it's going to create lots of heat because all that energy will be wasted. So I was interested first to see how I can actually activate or stimulate this system, and if there is any way that we can monitor them non-invasively using an imaging modality. So we had micro PET, micro CT at the center that I used to work at UC Irvine, and we decided to go with the activation, which goes with the metabolism. So we basically, these series of experiments, we use F18-FDG. FDG is an analog of glucose labeled with fluoride 18. And this shows that the metabolism in that cell goes up. This is like very common radioactive analog of glucose that it's used for finding metastases for cancer as well. So what happens during the search that I was doing at the beginning, I found that there are lots of articles that they talk about activating brown fat based on cold temperature. And the mechanism of that cold temperature is through adrenergic receptors. But there are several type of adrenergic receptors like beta-1, beta-2, beta-3, alpha-1, alpha-2. So, and then I searched to see what type of receptor we have more on the surface of these brown adipocytes. And I found that we have more beta-3 adrenoreceptors here. Obviously, norepinephrine activates these receptor, but norepinephrine activates beta-1 and beta-2 and the others. So it will be lots of cardiovascular pulmonary side effect if you use norepinephrine. So I had to use something which is very specific, a very specific agonist for beta-3 to have like a specific effect. There were lots of papers that they used nicotine, caffeine, ephedrine, norepinephrine, which directly and indirectly stimulate these noradrenergic receptors. But then I found this medication or this compound known as CL316243, which was an agonist for beta-3 with very high affinity to beta-3 adrenoreceptor. And then what it does, because the beta-3 adrenoreceptor is a G-coupled protein receptors, it's going to activate adenylyl cyclase, which converts ATP to cyclic AMP and goes through the protein kinase A and increase the amount of acetyl-CoA in the mitochondria, and that cause more of the hydrogen ion forced to go to inner membrane space of the mitochondria. On the side, when the assumption of the free fatty acid increases in the cell, the intake of glucose also goes up because the cell needs to compensate that extra metabolism that it's going through. So for that reason, there will be lots of glucose uptake through glucose transporter. So this gives us a good opportunity that we use F18-FDG. If you remember, or you have learned about glycolysis, the first step of glycolysis is to phosphorylate glucose and converts glucose to glucose 6-phosphate. So FDG also gets this 6-phosphate because phosphate is charged. This will be trapped inside the cell and we accumulate this. So we see that under PET scan that this tissue gets hotter and hotter because of this accumulation. So let's go to the next slide. Okay, so in the first experiment that I did, I wanted to see if the CL316 is as effective as cold. So I compared it to the cold exposure. And then also we know that this is beta-3 adrenoreceptor, but just to prove that the mechanism is the same mechanism as a control, I also used that the drug plus propranolol, which is a beta blocker, to see if that blocks the effect or not. So I used male Sprague dolly rats, and I had a group of these rats exposed to the cold temperature, about 8 degrees Celsius, for about 2 hours before PET imaging scan, and then injecting them with another group with the CL316, and then do a PET CT scan on them. So the way that we did the PET CT scan was after the injection of the CL316, we waited for 30 minutes. Cold exposure was for 2 hours. Then we inject them with F18-FDG. We waited for about an hour, 60 minutes, so for the uptake to happen. And then we run the PET CT scan for 30 minutes PET acquisition, followed by about 10 minutes of CT acquisition for attenuation correction and anatomical delineation of the PET images. So what you see here, that this is known as interscapular brown adipose tissue. So you see that in cold, we can see that it's getting activated as compared to the control. So this will be the cold exposure, this will be control, but it's not that a big difference, right? So 2 hours might not be good enough. So some articles, they did like 6 to 8 hours. But when we inject CL316, you see that the effect is completely higher, and it activates brown fat. We added propranolol when we inject the CL316 in another group, and we saw that the effect was gone. So this beta-3 adrenoreceptor agonist help us actually to show where the brown adipose tissue is in the rat, because at the time that I did this experiment, there was no mapping of the brown fat available. So we could find like a cervical brown fat, interscapular brown fat, lots of brown fats around aorta, periaortic, that explain why our central body temperature is higher, and then intercostal brown fat as well that you can see here. So we quantified this, and you see this quantification, which is significantly different than the other groups in terms of the F18-FDG uptake in the IBAT. So next, what I thought about was, okay, so we have these beta-3 adrenoreceptors. We activate these brown fats with CL316. I knew that if I inject norepinephrine, that's gonna also activate it. But the problem was that the exogenous norepinephrine may kill the rat, and it's not healthy, even if like it's not translatable to clinic. So then I thought about, I searched a little bit more, and then I got familiar with something called NET, which is norepinephrine reuptake transporter. So in a synapse or a junction, when norepinephrine is released from the end of the axons or the presynaptic membrane of sympathetic neurons, then it goes to this synaptic cleft, and then it's going to bind to the beta-3 adrenoreceptor. But then there is a mechanism that they need to be reuptaken to go back to the end of the neuron. And that's through the net. Now, my idea was that if I can block this net so it cannot be reuptaken, then the concentration of this norepinephrine will get high here, and then there will be more stimulation of this. At least this was endogenous, so I thought that the side effect will be less if we do this. Then I found, I searched about like medications that they may actually block NET. I came up with a medication called atomoxetine. Atomoxetine is a medication which is used for ADHD, treatment of ADHD, which blocks NETs in the frontal cortex of kids that they cannot focus for study. So, and because we have the same mechanism, the same type of synapse that we have in the frontal cortex in brown fat with the innervation of the adrenergic neurons, then I thought that if I block those NET with atomoxetine, I should be able to see the effect. So then we designed the experiment and we did it. First, we did this on the same Sprague-Dawley rats, not the same rats, but the same species, Sprague-Dawleys. And then the rats were fasted, and we injected atomoxetine, waited for 30 minutes, and then injected F18-FDG, waited for an hour, and then run CT scan, PET CT scan with the same protocol that I mentioned. And we saw that also atomoxetine, basically, here you see just PET, and this is PET and CT merged together. That activates brown fat. You see that the cervical brown fat, periaortic brown fat, they're all activated, as well as the interscapular brown fat. When we used propanol, all those activation gone, so there was no activation. So this shows the same kind of mechanism through beta-3 receptors or through adrenergic pathway as compared to control. So we quantified them, and we saw significant difference between atomoxetine and the other two control groups. And after the experiment was done, the rats were sacrificed and the brown fat was removed. And then we basically made slides of these brown fat and exposed them to film. And the consecutive slides, just we got a photo of that to show that where the brown fat is. So when you remove these brown fat, they're actually brown because of like the high mitochondria they have. They're not like yellowish or whitish that you see on white adipose tissue. So, and we can see that it's the amount of the activity basically matches to the anatomy of the brown fat that they were here at atomoxetine. When it was with propanol, you see exactly like the same structure. The activity was kind of less as compared to control. So this also showed us that blocking NET increases that norepinephrine concentration in there, and that cause activation of brown fat as well. Then what happened, I also did some imaging on non-fasted animals. So one of the very common problem that scientists have working with F18-FDG is that the blood glucose level can change the way that F18-FDG is absorbed in the target organ, because this is all based on competition. So if a rat is not fasted, then there will be lots of insulin available in the blood because it's postprandial, and that insulin may push glucose to go to the other tissues. And then by that, F18-FDG goes down, and because the amount of the F18-FDG is limited, any organ that can absorb more of that glucose because perhaps they have more glucose transporter, they're going to get more of the uptake and the others will not be able to do it. So in a control group, we saw that when we inject the F18-FDG, we see lots of accumulation around heart or in the cardiac muscle, which makes sense. Like brain and cardiac muscles, they got like the highest amount of F18-FDG activity. And that's because it's postprandial. So this could be a little bit problem if we want to translate this to the clinic. Do we need to ask people to have the medication just fasted, or can they take it at any time during the day? So for that reason, when I did on non-fasted rat, this experiment on non-fasted rat, I actually and like our protocol was just wait for 30 minutes after the medication and then inject F18-FDG. Here I actually did 2 time points, 30 minutes and 120 minutes, and compare it with that 30 minutes that originally we had for the CL3116. And I saw that if we wait a little bit longer for atomoxetine, like 120 minutes, then we can see the activity more consistent as compared to 30 minutes. Because in the competition between like hearts, brains, and brown fat, atomoxetine is not that strong, so it takes some time to pick up some of the F18-FDG. But that was not the case for CL3116, because that's like a very high affinity compound for beta-3 adrenal receptor. So for that reason, could just suck the whole carbohydrates and fats into the brown adipose tissue. So when I saw this, then I thought that, oh, so this is like a static image. Like we wait for about 1 hour after the uptake happens, and then we just run PET for 30 minutes to get one image, and then 10 minutes after. What if we run a dynamic PET image for 120 minutes long PET scan, and then having 10 minutes CT scan after, and then we inject while the rat is under the PET scan, and then we can actually see how like the pharmacokinetic basically of the F18-FDG in there. So this is what you see here. So basically we ran the PET for two hours, and then we injected after we start the PET scan. And so F18-FDG was injected at this point. And we saw that in the group with atomoxetine, the uptake is really fast even at the beginning, and it remains at higher. So the clearance of this will be lower as compared to control group. So the SUV standard uptake value was significantly higher in A, B, and C. A was like about 5 minutes after injection and PET scan started. B was about 10 minutes, and then C 30 minutes, and then remain higher consistently after that 30 minutes. So I wanted to see how, like what's the difference in terms of the clearance rate of the F18-FDG from the IBAT or interscapular BAT in the absence and presence of the atomoxetine. So what I did, I got this data, and then from C, as you see here, and I calculated the logarithm of the value, the average of every 5 minutes divided by the value at 30 minutes time. So which will be starting here. This is at 30 minutes, and then going 5 minutes interval until 120 minutes, and calculated their K off, which is that clearance rate. And we saw that without atomoxetine, this clearance rate is much, much faster than clearance rate when atomoxetine is there. So looks like that, you know, atomoxetine being there kind of pushes this beta-3 adrenal receptor to work for a longer time, and then let more F18-FDG goes to the brown fat. So there are some articles also that they show that they, in a clinical setting, I think it was in 15 women that they did, they used atomoxetine, and they saw a little bit of weight loss. They did not mention that it was through brown fat, but I guess that it could be through brown fat. but that weight loss returned back to normal after 2 weeks, I guess, if I remember it right. So all these data that I showed you were on rat. Then we thought that, let's do this on mice to see if we get the same result. So we did the experiment on male Swiss Webster mice, and we used CL316. And we saw that in mice, like they are smaller, PET scan could be a little bit tricky for them, but we found that IBAT still gets really hot in mice, and it's very obvious. So we chose to compare IBAT uptake from this point when we do mice because of its more consistent shape. So it was really easy to just draw ROI around it. So for that reason, we just moved through just comparing IBATs between different groups rather than getting like different parts. Although you can see some like peri-aortic, or some other places like cervical, but it will be difficult to draw ROI around them. So so far, so if we consider this as the cell membrane of brown adipose tissue, this is the presynaptic membrane of the innervated neuron. So far we talked about activating beta-3 adrenal receptors with these agonists on this membrane. We also talked about atomoxetine, blocking that and increasing norepinephrine here. But also we briefly talk about how these beta-3 receptor works, which is a G-coupled protein, and then it activates an enzyme, which is adenylyl cyclase enzyme, and that converts ATP to cyclic AMP, which is a secondary messenger, and run the whole cascade of the pathways down to waste the hydrogen ion. So I also found another medication, forskolin, which is in the market as well. And forskolin directly bypass all this and directly activate acetyl cyclase. So adenyl cyclase, sorry. So and then when this is activated, my assumption that the cyclic AMP goes up. So and then we are going to have the effect. And we did all these three in the next experiment in mice because the previous data was on rats. So we thought that, let's do all these three in mice, see if we get the same consistent result. So here you can see the result, like with CL, definitely the way that we see it in the rat, we can see hot spots of the intrascapular bat. With atomoxetine, as we expected, it was still active, was less. Forskolin was kind of the same thing. So activated was, but was less than CL. And this is like compared to normal. We quantified them and we saw the significant difference between CL and control, and also significant difference between CL and atomoxetine and forskolin. So significantly has better effect because it's beta-3 adrenal receptor agonists with very high affinity. And also both atomoxetine and forskolin had significant differences compared to control. So all of them show that they can activate brown fat, and we were able to image these activation. The thing that we learned from here, because we get different level of activation, so we can call it like severe activation or moderate activation. So it kind of gives us a range. So this gives us lots of tools to actually work about it. One thing that I was interested that we were taking CT scan and we just using CT scan for correction and delineation of the anatomy. Then I thought that, why not we use the Hounsfield unit of the CT scan and see if there is any effect in there too. So in the original experiment that we did, although there were some changes, but because their standard deviations were like larger than expected, we couldn't see that much of significant difference in the Hounsfield unit. But then later on, when we improved the techniques and all stuff, so this was not the case. So I found that when I activate BAT, and especially when it's with CL, like it's severe, as you see here, as compared to mild activation, I can see, and these are the CT scan, but these are the inverse CT scan. So instead of a black background with a white image, you have like a darker image with a white background to be able to see the effect. So so basically this white area here, it's not denser, it's darker. In a real CT that we usually used to see. So I saw that, you know, the Hounsfield units are much lower when the brown fat is not active or it's mild activation, and it becomes darker, like at the level of the tissue Hounsfield unit when it gets activated, which is very interesting. So we know that the Hounsfield unit of fat is around -150 to about -50. Air is like -1000, and tissues usually 0 to about 50. Bones go to 1000, sometimes 2000 Hounsfield unit. So, so this means that if the Hounsfield unit moves from -150 to -50 or 0, looks like that the fat, this means we can translate this, that the fat is gone. So lipolysis happened. So there was like a fat loss in that area, which basically makes sense when you activate brown fat. All those fat droplets that they are in there, plus the fats that are around them, like in the white adipose tissue, that they have gone through lipolysis. So the amount of them are less. Now tissue has higher Hounsfield unit. So basically we plot the standard optic value of the IBAT against the Hounsfield unit of the IBAT. And it was interesting that as SUV or the uptake increases in PET, the Hounsfield unit also increases. The R square was about 0.55, and it was significantly different. And also it was interesting that I saw the same kind of effect when as compare the Hounsfield unit of IBAT. So basically now with this, I could prove that, you know, lipolysis happening in IBAT. Now using this one, okay, lipolysis happen in IBAT. Is there any other places in the body that we have lipolysis? Because cannot be just localized, like this is like a general thing. So do we have other place that we can work on, like we can see that this happens? So if you have seen CT scan for a fatty liver, you see that the Hounsfield unit gets lower and lower as fat accumulated in the liver. So I saw that in the Hounsfield unit of the liver of these mice, it actually increases when the Hounsfield unit of the brown fat increases. Means that as we have lipolysis in brown fat, we have lipolysis in liver as well, which kind of implied in my mind that this medication or this approach could be a good approach for fatty liver disease, non-alcoholic fatty liver disease or alcoholic fatty liver disease. This can be useful. Also, we know from some other articles that brown fats, they basically, when they start gets activated, we may lose weight. We lose weight by breaking the fat that are stored in the white adipose tissue. So here you can see also there was a correlation, a nice correlation, a strong correlation between the SUV of the IBAT and the SUVI, the white adipose tissue, which is around that IBAT, and which R square of about 0.5. This also shows that when you activate brown fat, it's not just the brown fat, it's going to activate white fat. After I published, there were lots of papers that came out that they talk about beige fat and browning, which means that white adipose tissue can become brown adipose tissue, and in the process they become beige. So this is something that I think that happens when you start activating brown fat. It also stimulates white adipose tissue directly or indirectly. I'm not sure about the exact mechanism. We also see there was a relationship between the muscle SUV and the IBAT SUV. So as we have more glucose uptake in the IBAT, there was more glucose uptake. Come on. There was more glucose uptake in the muscle, which basically tells us that it may actually increases the resting metabolic rate general, not just in brown adipose tissue or white adipose tissue. So muscle also start to uptake more of the glucose. So we couldn't see any significant difference between heart uptake. So we can say that the medication is safe. So it's not going to increase the heart rate or heart activity. There was no relationship between uptake of liver or spleen to the brown adipose tissue as well. But the uptake of brain went down. There was like a reverse relationship, which makes sense. It's, as I said, it's a competition of the glucose. So if brown fat uptake more glucose, there will be less glucose available for brain to be used. This could be a side effect. It may cause some of the symptoms of hypoglycemia that we can see in diabetic patients. So this is something that we need to consider if you want to translate this to clinic. So, so far I just talked about brown adipose tissue. I want to switch to the pancreatic islets. So for the brown adipose tissue, the approach that we used was doing a functional imaging, basically based on F18-FDG. We activate the cell or the tissue, and then they start to burn more glucose, so they need to uptake more glucose, and we just measure that uptake, which is basically the approach that if you use F18-FDG in any place, that's the thing that happens. But for pancreatic beta cells, we use another approach. Instead of working on their function, we found that in one of the articles published in 2005, we found that there are dopamine receptors on pancreatic beta cells. There are 5 of them, D1 to D5. So we have D2 receptors that they're expressed in pancreatic beta cells, dopamine receptor subtype 2. And in this article, they did all rat, mouse, and human, and they stained them for the insulin. Then they stained them for the D2 receptors, and they overlay, and they saw that basically wherever there is insulin, whatever cell, we have D2 receptors. So there's strong belief that these D2 receptors are pancreatic beta cells, because these are the cells that they contain insulin in pancreatic beta cells. So the experiments that we designed was based on these facts that we found. So now, if that's true, if we can use an agonist or antagonist that binds to these D2 receptors, and then we can label it with something, and we can detect it, we can use it for different approaches. Could be in patients with diabetes to see their progress. We can use it as a screening imaging for if someone is losing pancreatic beta cells over time, if they have the risk factors. They are not diabetic yet, but the number of the pancreatic beta cells going down over years, because it's a chronic process, and we might be able to prevent. So we can actually see those pancreatic beta cells in a normal pancreas. So these are like the applications that we thought about. So we designed the experiments based on that as well. So first of all, we used a compound called falipride, and falipride is a D2D3 receptor agonist, and it's the affinity to D2 is much higher than D3. So then we started to work on the rat islets to see pancreatic islets, which they have beta cells and alpha cells, to see if they bind to them. So the first experiment that you see here is to kind of see if the binding of the falipride to these D2 receptors specific to the endocrine part of the pancreas or exocrine part of the pancreas. Pancreas is an organ with two parts in it. 98% of the pancreas is exocrine and just 2% is about endocrine, which is the pancreatic islets that contain the alpha cells and the beta cells. So what we did is that we basically isolated. So there is a process called pancreas isolation or pancreatic islet isolation. So we can, in a process of like 3 to 4 hours, we can isolate those pancreatic islets from the whole pancreas. So we did this, and then we stain them. We kind of separate the endocrine from exocrine. We also stain them to see if they're alive or dead in the culture media. And then we expose them with the falipride, and we measure the falipride uptake. And this is the quantification of it, that endocrine uptake was much higher than exocrine uptake. We put these cells, we fix them, and then make slides on it, and then we, without any labeling, and then we label them after that when we fix them. And you can see them here that they get the falipride. To just make sure that this is not a non-specific binding, we also did it with haloperidol, which is competitive with falipride and binds to the D2 receptor. So it removes the falipride. And we saw that the binding is removed. So the binding to D2 receptor was specific. We also wanted to see how PET scan can detect this very low number, about 50 islets equivalents of the cells here, if they can pick up the signal. So we put this slide under PET scan, and we saw that there is a difference between two. So the PET can determine the difference of even like as low as 50 islets equivalent. Then what happened, we wanted to do a PET CT scan in a normal pancreas. So we basically used the falipride. We let the falipride binds, and then we we did the PET CT scan. We run the PET CT scan for about 60 to 90 minutes. That was also like a dynamic PET-C, how much they uptake, how much they retain. And then we use different parts to basically control, like as a control. So here in a PET overlay of a CT of a rat, you can see the spleen. The pancreas is between the spleen and the duodenum, so it's attached to the duodenum. So you can see stomach at the top of it. So this will be the liver, and these are the two kidneys. So when we look at different areas, so we saw that when we use haloperidol, so basically the effect gets less, or let's say it this way. So in a pancreas, when we looked at the pancreas, in a control pancreas and in rats that we actually injected the falipride. So you see that in a falipride, the uptake goes high and then remains at the higher level. When it's injected with falipride, and then it goes up, but then it falls off from the D2 receptors because of that competition. So we use the erector spina muscle, which is in the back. So kind of safe, being far from any different hot spots. So, and we saw that in both cases, there is no significant difference between these two groups. So this shows that these, even like in a normal endogenous pancreas, we can actually see that there will be more uptake in the pancreas because of the presence of the D2 receptor. Then what we did, we actually sacrificed those rats, and we took the rats and put just the rat under the PET scan, and we measured that, and we could actually see the difference as well without haloperidol and with haloperidol, that the activity went down. This is like after the PET scan is done, the rats are sacrificed and the tissue is procured, fixed, and then put it under the microscope, under PET scan for the measurement. Then we thought that, okay, so what happens if we actually make our animals to become diabetic? So what we did is that we actually used streptozocin and induced diabetes in this animal. So here you can see the immune staining for the pancreatic tissue for the animals, that you can see all these pancreatic islets that they are brown. After diabetes, all these pancreatic islets are gone. So when we inject these control and diabetic rats with falipride, and then we procured falipride, we could see the same effect of the falipride activity in control and diabetic. So it basically decreases the number of the amount of the activity of falipride due to the loss of the pancreatic beta cells. Then the next question was that, can we actually monitor the transplantation of the pancreatic islets into the rat's body? So we did it in three different ways. The first way, which you can see here, we isolated these pancreatic islets from rats. Then in vitro, we added falipride, incubated them with falipride, and then we washed them, and then we injected pre-labeled islets with the falipride. So you can see that this is the place that we transplant them in the spleen, and as a control, we just transplanted splenocytes with the falipride. So the splenocytes, they were not able to retain the falipride, so you don't see that much of activity. And then here you can see these pre-labeled islets in there. So that was one activity. The second one was, let's just do the PET scan without CT scan, because it might be tricky if we don't know where spleen is. So we are transplanting these islets into spleen, and let's not pre-label them. See, after we transplant these rats, if we inject the falipride into the tail vein of these rats, these pancreatic islets uptake. So for that reason, to just make sure that the area I'm looking is the area of the interest, I transplanted these pancreatic islets in the spleen, like in the cortex of the spleen. And then I basically sutured the spleen to the skin, so I know where the spleen is without using any CT scan. So, and then I did the PET scan, and I saw that these pancreatic islets actually were able to uptake the falipride. So this also shows the result, and you see the quantification. So it goes up and it stays at the higher level in the spleen after transplantation, as compared to the control of a muscle. Then I found a contrast for a spleen, which could, like after injecting that, this is like a CT scan contrast for spleen. And I injected the rat with that, so we can actually see the spleen. I don't need to manipulate the anatomy of the spleen to be able to make sure that the area that I am looking at is the right area. So I transplanted the islets into the spleen. I injected with the CT contrast and injected with falipride. And again, I saw that in the transplanted spleen, there will be more uptake as compared to the control spleen, or the muscle in both cases. So this shows that also we can use a tracer that they just bind. It's not functional. We are not measuring the function of these cells, but if the cells exist in that area, they can uptake that agonist or antagonist, and we can label that. And I think that will be it. So thank you so much, and I'm available if there is any question.
Can you hear me or not?
Now I can hear you. I see in the chat there is just one question asking for my email. You can contact me through Reza M, R-E-Z-A-M, at uci.edu. I cannot hear you. You're mute.
Hear me now?
Yes.
Yep. So there's a question that has been asked by someone called Kimia. She asked multiple questions, actually. First is, with regard to FDG PET imaging, is there any alternative sort of radio tracer to perform similar type of studies?
Alternative means in terms of the radioactive, like C11 or something like that?
I presume maybe radiopharmaceutical in a sense, maybe something else other than glucose to image sort of metabolism, maybe, or in your case.
I've not worked with it because F18 FDG is very common here. It's cheap. You can get it from anywhere around. So there might be. And the PET is just one modality. We have like SPECT and other stuff that you can use. Even there are some tracers that you can use for CT scan as well, in terms of the anatomy. I'm not sure about function, but if you want to see the structure, you might be able to use them.
Cool, thank you very much. Another question again asked by Kimia. She was asking regarding the sort of NET studies that you've performed, and she said, what is the purpose to increase the accumulation of NET that you use the ATX drug?
So the thing is that in any synapse, when the neurotransmitters is released, they need to be reuptaken. You cannot have like the effect forever on a receptor. Like receptors by nature is like human. So if I just keep talking for 3 hours, 4 hours, you're going to shut down your camera and go home, right? You cannot tolerate that much of information. So the same thing happens to the receptors. So if you have constant stimulation by a compound, the receptors shut down and the cell starts to downgrade all those receptors. So the number go down. So the effect will be less. One example is like losing hair. If in men, high testosterone, you're going to lose, in people that they're bald, they may have high testosterone and they may have low testosterone. In both cases, they're going to lose hair. It's because even high testosterone is going to downgrade the receptor. So that's like the nature of the receptor. So for that purpose, there are always a mechanism that removes those neurotransmitters. The receptors can refresh themselves and then they secrete it again. So this should be pulsatile. So NET is basically doing that. So the norepinephrine binds back to those transporter and then gets to the end of the axons of the neurons to be able to go back again to the synapse. So now if you block it, you're going to have these pulsatile less, but will be a little bit more effect. This effect will be temporary. You remember I said that there were like a study on 15 women for losing weight. So when they use atomoxetine for them, they lost weight for 2 weeks, but then after that stop. That's the place that the receptors start saying, hey, I'm not going to listen to you anymore. So you can do this to activate. For mine, it was like 1 hour, 2 hours. That's not a big problem. So they listen. But if I keep doing it, then they become resistant and there will be less effect after a while.
Cool, thank you very much. Following this point, there's another question, final question asked by Kimia, and she was asking, what is the aim of brown fat activation?
So whenever you activate brown fat, because it uses glucose and fats, and first it used the internal glucose and fat, and the purpose of using them is to burning them, going through glycolysis, Krebs cycle, electron transport chain to produce ATP. But the mitochondria in brown fat has that protein called UCP1, uncoupling protein 1, which wastes all the effort that cell does from the glycolysis to the electron transport chain. Because if, let's say that you eat something and you get the ATP, then the ATP that you get, the extra that you ate will be stored because your body now has enough energy for the moment. And whatever is extra, send a signal to these cells that, hey, this is extra. I don't want this extra because I have enough ATP. So go and store it so you gain weight. So if you put a mechanism like UCP1 to waste the hydrogen ion, so although these carbohydrates, sugar, and fats, they basically get burned or get oxidized to the level of hydrogen ion, but ATP is not synthesized. So the cell will be short for ATP synthesis constantly. Although burning all these carbohydrates in a high concentration, it's not going to give the cell the ATP required because it's wasted. So it starts burning. As it starts burning, then the resources in the cell will be short, starts to send a signal to the whole body. So you're going to start to do lipolysis in your white adipose tissue. You lose weight because the fat is gone. For fatty livers, the fat in the hepatocytes will be gone to the lipolysis. Glycogen storage in the liver will be depleted. So it basically sucks all the blood glucose and fatty acids to basically burn, because the cell's telling them, hey, although you are doing all this work, I'm not getting the ATP I want.
Cool, thank you very much. There's another question that came out. They're referring that, so you've used FDG, and that's glucose imaging. And how did you sort of control for the exercise or physical activities of the subjects before actually performing the scan? How did you sort of normalize them? So when, with regards to animal handling, so maybe they could have some sort of FDG uptake as well in the sort of IBAT area as well.
That's true. So we have the animals like in an area which is controlled on a 12-hour light dark cycle. So that was controlled in terms of the activity. All the PET scan and FDG injection done by me, just one person. So it was not like different people that they do it. Handling was not an issue. And then the room was always like maintained in a room temperature. It was done in a room that we had the PET CT scan, which we usually keep it cooler than in a normal room for the equipment. All of the injections were done under anesthesia, so there was no pain that kind of increased their heart rate and all the stuff while we injected. And then during the after F18 injection, waiting for 1 hour, I did not wake up the rat. So I maintained anesthesia. So the physical activity during that hour was kind of the same for all of the animals.
Thank you very much. And were you interested to actually sort of perform all these studies that you've mentioned with regards to the adipose tissue in a sort of physical exercise model as well, to see the effect of exercise rather than just effect of drugs?
I know, like in my experience, I know that even if you want to focus just on drugs, it's not going to work, because the brown adipose tissue or weight loss, if the purpose is weight loss, is a multifactorial thing. So you can't just have, okay, so have this drug and it's going to help you. But if you combine this with exercise, definitely the effect will be synergistic effect. So it has like higher level of effect. I did not do any experiment on it, but I have read about it from other colleagues who have worked on this. And I know that for sure exercise also activate brown fat.
Cool, Thank you very much. You mentioned SUV. So do you mean by in silico sort of SUV or ex vivo biodistribution?
So it's the distribution. So it's a standardized uptake value that when I draw the ROI, so there was a protocol of like where we actually draw the ROI, we put the numbers in and it automatically, the system draw the ROI for it. So region of interest. And that was based on the edge of how much of like if you draw the map of the ROI in an area, you see in one area, all of a sudden you get a peak. And then we started from that peak to draw the border. And then we had that uptake value over there.
Cool, thank you. There are some more questions by Dr. Hassanzadeh. He or she is asking, firstly, they thank you for your great talk and this great webinar. And the first question he or she is asking is, the PET scans that you performed, were they sort of dedicated animal PET imaging, or they were human sort of clinical?
No, animal.
Okay.
Animal, small Invion. I don't remember exactly the model, but animal PET scan. All the parameters that are in my papers that I cited here.
Mm-hmm. And following the question is, apart from the medication, do you have any idea about nutrient sources of BAS activation?
I do, and I did not get a grant for a clinical trial here, but I actually did that experiment on myself with like a big difference. So I used to teach for also acupunctures and Chinese Oriental medicine students, so I'm a little bit familiar with herbs and like a non-traditional or Western medicine. And I actually work on ginger. And I found that ginger activates brown fat as well. I injected rats with ginger. Then I started to eat ginger myself and I saw the effect, like my blood glucose went down, HDL went up, LDL triglyceride went down over like 6 months of use. So I know for sure that that's also there. I had a colleague who was from Taiwan and gave me like a combination of 4 herbs that we saw, we injected in rat and we saw that those activate, but I never asked for formulation. So I don't know which herbs they used, but I know for sure there are some. I think turmeric is another one that may actually activate brown fat as well.
Mm-hmm. Thank you very much. Again, Kimi has asked another question, but I don't know to which part, maybe you can understand. So she's asking, so this mechanism is used in diabetes while the patient is losing weight. Is that right?
Yeah, so because when you activate brown fat, it activates both lipolysis and consumption of carbohydrate. So it can bring down blood glucose at the same time cause weight loss. So assuming the patient has brown fat, so you can activate them. There are some people that they may not have brown fat, so you may not see any effect. If your patient is diabetics, I do not consider giving them, let's say, ginger or something as extra beside their medication, unless you are a medical doctor and they're doing it under your supervision. Otherwise, it might be a little bit difficult to manage. If someone is injecting insulin at the same time you activate their brown fat, you may actually cause hypoglycemia.
Mm-hmm. Thank you very much. Last question by Dr. Hassanzadeh. He or she's asking you to share your experience regarding tissue scans, which probably I presume that he or she means autoradiography and the ex vivo sort of PET imagings that you performed. How was your experience using them? Which one you found more interesting or sort of more sensitive in a sense?
They were just confirmatory. So whenever we want to like do an experiment in a larger scale that will cost more, first we need to have like a proof of concept. So if that works or not. So usually injecting with a tracer, sacrificing the rat, get the tissue out and put it under PET, or doing like autoradiography is not that expensive. You can do it like in a day. So that is the concept rather than like a 3 months, 4 months, because all these groups that when I said like we had like a group of 5 rats, we did this PET scan. Each of these rats PET scan was done in 1 day. It was not that you do all, because it's just one PET scan. So every day you do just one and then you put them together. So it's a long process.
Mm-hmm. Cool. Thank you very much. Just to let you know and to make you sort of interested as well, or maybe proud, as you said in the beginning. So we have actually performed some brown adipose tissue imaging as well using FDG in our core facility. So because we are a sort of a CRO, we're not sort of research center in a sense, but there were, as far as I remember, in the last 2 years of our lifetime, we've had there was a sort of a group, I can't remember from which university, they did actually perform some of these imaging, and it's interesting to sort of continue this as further collaboration between yourself and scientists here in Iran as well. So
Yeah,
I'm looking
definitely.
forward. Yeah, and we actually have our sort of annual symposiums. So we will be this one will be our fourth annual symposium, and will be happening in summer. And we would love to have you as one of our speakers as well, in case if you have the time.
Anytime. Yeah, I'm really glad to see that this is happening. Last time I was in Iran, it was 10 years ago, like 2010. And I remember that my dad's friend wanted to bring his wife's PET scan. So I look at it and I asked them, do we have PET scan in Iran? I did not know that. And then when the guy brought it, I saw that they went to Istanbul and did PET scan. I was like, oh my God, why should our patients go to Istanbul and do PET scan? So there was no PET scan. I don't know if there is a clinical PET scan right now in there, so it's active, but there was nothing over there. I was like, this should change. So I'm really glad, and thank you so much for doing it.
Thank you. So just to let you know, I think roughly we have around 5 clinical PET scanners in all Iran at the moment, active and running. But the PET scanner that we have is dedicated small animal PET imaging.
Mm-hmm.
And just to make you more even interested and proud as an Iranian, is actually homemade. So it's all built.
That's cool.
Yeah. So the imaging devices that we have in our TPCF, all of them are made in Iran and made by Iranian scientists and engineers. And even some of them actually have US patents on them as well. So it's not like a reverse engineering kind of
Cool.
scenario.
I'm so proud. You gave me goosebumps.
Yeah, thank you. Yeah, really looking forward to have you here in Iran, hopefully sometime soon, or even until then, maybe we can have, as I said, have you in our symposiums, or
Sure.
I'll send you a sort of a presentation in Farsi as well regarding TPCF,
Okay.
just for your interest and make you more proud as an Iranian.
Yeah, thank you so much.
Thank you very much. It was an honor to have you, Professor Mirbolooki, and I really enjoyed your beautiful storytelling as well as drawing as well. That was a good mixture of drawings and sort of storytelling of your science.
I give the credit for this to COVID. So during the last year, I was just teaching online. So basically improve my skill in online education.
That's great. So, and I wish you very in advance sort of Happy New Year and a Merry Christmas as well.
Thank you so much.
Sort of Iranian Yalda Mubarak to you as well.
Thank you, and have a good day.
Thank you very much.
Thank you, everyone.
With that, I would end the session, and once again, sincerely thank Professor Mirbolooki for his amazing talk and for his time to share his beautiful research with us. And we would I would ask actually our admin to present the poster for our next sort of webinar, which we are having in a month's time. So as you can see, we would be having Dr. Jack Sharkey, and he's at Liverpool Preclinical Imaging Center, where his talk would be talking about sort of the preclinical imaging facility at University of Liverpool in England, and some of their case studies as well. So with that, I would like to end the session, and once again, thank Professor Mirbolooki for his time and for his great talk. Thank you very much.
Thank you. Have a good day.
Thank you, you too as well. Bye.
Bye.